All right, the first part that we're going to go through in Chapter 10, which is called Gene Action from DNA to Protein, really kind of picks up where we left off with Chapter 9 with DNA and DNA replication. DNA holds the code for making proteins. In this chapter, we're going to actually talk how those proteins are made. So in this first section, our three big learning outcomes are that you're going to really understand and be able to draw the central dogma. You're going to be able to list the three major types of RNA and their functions, and you're going to compare and contrast DNA and RNA, our two nucleic acids. When we talk about gene expression, we're really talking about, that gene, that's that segment of DNA that codes for a protein. We're actually talking about getting it to the point where it actually is expressed as some sort of trait, meaning you have to have that protein being made in order for that trait to show up, such as blue eyes or brown hair. Well, one of the terms we probably didn't cover very much in biology is what's called the exome. And the exome is the part of your DNA in your human genome that actually codes for protein. Okay? It only represents 1.5% of your overall genome. So that's kind of an astounding number, that only 1.5% of your DNA actually codes for proteins. So what is the rest of it there for? Well, quite a bit of it, much of the human genome, controls protein synthesis. So it doesn't actually code for proteins, but it plays some role in controlling the process of actually making the protein. And then we do have some DNA that doesn't, maybe doesn't code for anything. That can kind of provide us with some protection against mutations, because obviously if mutations happen, odds are that it's going to happen in a non-coding region that doesn't code for protein. And just a little fact here, your genes encode over 20,000 different types of proteins. So that's kind of an interesting number as well. You don't need to remember that number. That's just for reference. And just for review, your proteins are very, very important. That's why this process that we're going to be talking about in this chapter is so important. This is just a small snapshot of just a few of the proteins you have in your body and what functions they play. And so your body is made up mostly of all those different kinds of proteins, whether they be enzymes or different structural proteins. And we definitely need to have them made in our body for us to function. So again, gene expression is getting to the point where you can produce that protein and thus have that trait from the instructions on the DNA. Okay? It generally involves these two main processes, transcription and translation. Translation is where we copy the code from DNA onto an RNA molecule, and then translation is using the information on RNA to make a protein. Okay? And that happens by lining up the different amino acids in a specific order. Now, even though this isn't involved in these two processes, transcription, translation, once we actually make the protein, it has to be folded in a particular form, specific 3D form. That's because, I'm going to put a star here, you can add this into your notes, the shape of the protein determines its function. Okay? Shape of the protein determines its function. So once we get done with transcription and translation, we actually have the protein made. It still has to do some specific folding so that it can actually function like it's supposed to. So the central dogma, a dogma is just like a big overriding kind of organizing, unifying principle. And in biology, the central dogma of molecular biology is very, very important because it shows the directional flow of your genetic information. In other words, it shows how your DNA codes for your traits. how it codes for your different proteins. It involves the processes of replication, transcription, and translation, and it involves the molecules of DNA, RNA, and proteins. So it shows you directionally how we go from DNA all the way to a protein. I'm a little more linear, so I'm going to show you another way to draw it that kind of also shows the flow in a kind of a flow chart format. Oops. So first we have DNA over here, and what we're trying to get to is proteins over here, right? Because the central dogma shows you how DNA codes for your proteins, right? So we know that DNA is able to replicate itself, right? So that arrow represents replication. Oops, I'm going to do a different color for the processes versus the molecules. So let's use blue here. But DNA is always found in the nucleus, and proteins are made on your ribosomes. So we have a little dilemma. DNA has a code, but it has to stay in the nucleus, and proteins are over here made on the ribosomes. We need a different molecule that's going to help us out, and that different molecule is RNA. Okay? The process of copying the code from DNA to RNA is called transcription. Okay? And where do you suppose that process takes place? Well, if we have to copy DNA, DNA can't leave the nucleus. This process also occurs in the nucleus. Okay? So replication occurs in the nucleus, transcription occurs in the nucleus. So RNA has the code, and it uses that code to help make the proteins. So I didn't need to put those that far away. The process we use to use the code to make a protein is called translation. Okay, translation. That occurs where again? Well, since we're making a protein, it has to occur on a ribosome. So you can see this one-directional flow here that shows you how your DNA codes for your proteins, and ultimately then that's what gives you your traits. So your DNA is actually your genotype, the specific coding that you get there, and then your proteins are what end up being your phenotype. If we think back to some of our terms from our genetic transmission unit. Okay? So if you forget everything else you learn in genetics, please remember this one thing, because you're going to have the central dogma over and over again in different biology courses, whether you take more biology here at West Fargo, or if you go on to college or any of those intro courses, they always cover the central dogma. So, two types of nucleic acids, excuse me. One is RNA, which is ribonucleic acid, and DNA, deoxyribonucleic acid. They both consist of nucleotides, meaning they're made up of sugar, phosphate group, and nitrogen base. They're joined, one after the other, just like you did when you made your DNA models. There are some differences, though. DNA is double-stranded. We call it a double helix because it's twisted in that twisted ladder-like shape. RNA is single-stranded. It still has a helix shape, but it is a single helix. Okay? It has thymine as a base for DNA. RNA has uracil. So U is going to bond with A, just like T bonded with A over here. Uracil just replaces the T. Both of them will still have adenine, guanine, and cytosine. Both DNA and RNA have those same bases. Deoxyribose is the sugar for DNA, thus the name. Ribose is the sugar for RNA. DNA contains the protein encoding information. RNA carries it to the ribosome, so it can be used to make a protein. Okay? DNA cannot function as an enzyme. RNA can function as an enzyme. And then DNA persists. That means it stays in the nucleus. RNA is transient. It can leave the nucleus. Okay? So make sure you know the differences and similarities between DNA and RNA. Again, they both kind of have that helical shape, but DNA is a double helix, RNA is a single helix. They're both made of sugars. It's just that DNA has deoxyribose. It's missing that one oxygen there that you can see over here on the ribose. And then notice that they both have C, A, and G. It's just that DNA has thymine, RNA has uracil. And the final part of this first part of the notes is the three major types of RNA. Messenger RNA, which we just abbreviate to mRNA, ribosomal RNA, which we abbreviate to rRNA, and transfer RNA that we abbreviate to tRNA. The names tell you what their function is, so really focus on that. Messenger RNA copies the code from DNA and brings it to the ribosome, like a messenger. It's carrying that code, it's carrying the message. Ribosomal RNA makes up the ribosomal subunits. Transfer RNA carries or transfers the amino acid to the messenger RNA on the ribosome. Okay, so each of them have particular jobs, but they're very similar to what their name is, so use that to help you. Again, carries the copied information from DNA that will specify a protein from the nucleus to the ribosome. A couple of terms here though. A codon is 3 amino bases in a row that will specify a particular amino acid. Okay, so every single codon codes for a particular amino acid. And that's what's used to build a protein. That's the subunit of a protein. The ribosomal RNA will help form your ribosomes. Ribosomes consist of 2 subunits. And here's these ribozymes. That's the kind of enzyme function that some RNA can have. Here is the large subunit of a ribosome and then the small subunit of a ribosome. And what the messenger RNA does is it kind of hooks on in between there and it brings its codons, and then those help the transfer RNA bring in their amino acids. So you can see right here, here's the messenger RNA, ribosomal RNA, and here's the transfer RNA bring in the amino acids to be placed here. And then transfer RNA has 2 ends to it. One of them is called an anticodon, and it's complementary to a messenger RNA codon. Okay? So if you see right here, here is AUG, GGA, UGU. These are all codons on the messenger RNA. And you notice that the anticodon is the exact opposite. It's UAC, CCU, ACA. Okay, they're exact complementary. The other end of the transfer RNA carries the amino acid. So here's the anticodon end, here's the amino acid end. This whole thing here, this kind of cloverleaf thing, is your tRNA. Okay, so here on this end, it's bringing it has the anticodon, so it knows where to match up on the messenger RNA. But on the other end, it's bringing the specific amino acid. Okay? And the thing to remember is that it's the codon that codes for that amino acid, not the anticodon. The codon UGU codes for cysteine. The codon GGA codes for glycine. The codon AUG codes for methionine. And we'll go through practicing that some more when we get to the genetic code.